speaker after coffee is Alfonso Mondragón, and he's going to talk about mechanistic studies of type 1A topoisomerases.
You have a pointer? I think it's on. Okay. Let me know if it's not loud enough. Do you have a pointer or I'll just use it? It's too high. How about this? Is that better? Oh, okay. Let me know if you can hear. But anyway, first let me thank the organizers for the privilege to come and give you a talk, and thank all you for your attendance. And it's been a very interesting meeting. I really enjoy and learn a lot of new things, a lot of things that I normally don't think about, and now I'm trying to think about. But what I'll tell you today about this, what we've been doing now for many, many years, about the mechanism of type 1 topoisomerases, in particular type 1A topoisomerases, and more about type 1A because they do more topological tricks than the type 1B. Okay, and I won't give a long introduction because Tony already talked about topoisomerases, but let me just show this slide to show a difference between what he showed and what type 1As can do. Topoisomerases can do all these reactions, but in these ones with single-stranded molecules, type 1A can also do these reactions. They cannot and are not single-stranded rings. They can link double-stranded rings. They can just link them, and if they are complementary, they can make it into a double-stranded molecule. So this can be done by type 2 enzymes. This can be done by type 1 enzymes, and you'll see in a few minutes that some of these reactions can also be done by type 1 enzymes. Now, there are, as Tony mentioned, there are two types of topoisomerases, type 1 and type 2. And an important difference is that type 1 breaks one single strand of DNA, okay? And in that way they change the linking number in steps of 1 or n. Okay? While type 2 enzymes break both strands of DNA and change the linking number in steps of 2. Okay? So these ones make transient single strand breaks, this one make transient double strand breaks, and that's a big difference between the two of them. Okay? Also mentioning what Tony said, type 1 enzymes do not require ATP for hydrolysis. They just use the energy stored in the supercoiled DNA for the reaction. So they don't need any external source of energy. Okay. Now the type 1 enzymes, they are subclassified to 3 types. Okay. Type 1A, type 1B, type 1C. I won't talk much about type 1B or not at all about type 1B or type 1C. I'll concentrate on type 1As. Okay. And what type 1As do is that they relax negatively supercoiled DNA. That's what they're known for. Okay. They require magnesium for activity, and they require single-stranded DNA regions for activity, and that's a very important requirement. That's the way they recognize which DNA to act on. It has to find a single-stranded DNA region for activity. Okay? So they sense the topology of DNA by sensing the presence of single-stranded DNA. Okay? All these other characteristics that they make a 5' covalent linkage, and that it's important, but not so important for today. They use an enzyme-rich strand passage mechanism. Okay? And unlike type 2 enzymes, when there are lots of drugs against them, there are no known inhibitors of type 1 enzymes, so there's no ones at the clinical level. Okay? Now, there are 2 mechanisms that type 1 enzymes can employ. Okay? One of them, okay, is enzyme-rich strand passage. The other one is swiveling. Okay? Type 1B and type 1C use swiveling or strand rotation. Okay? And that's a very straightforward mechanism. What you do is you form a neck, okay, and let the other strand, okay, they'll rotate or swivel around this other strand, okay, and in that way you can change relax either positively or negatively supercoiled DNA, okay, and you can turn many times or one time, so you can change the linking number steps of n, okay, so it depends how many times it goes wrong, okay, and these are fairly well studied. We understand more or less how they work. Okay. Type 1A enzymes, the ones we'll talk about, use an enzyme-rich strand passage mechanism. That's a similar mechanism conceptually to the one that type 2 enzymes use. And in this way, they form first they clip one strand of the DNA, okay, to form a gap in the DNA. And then they can pass the other strand through there, and they use it with using the enzyme as a bridge. So they pass another strand to the break. Then this enzyme passes, and then you reseal the break, okay, and then you have changed the linking number, and you change it strictly in steps of 1. Okay, so this is the mechanism that type 1A enzymes use. And if you think about it, it's very similar to the type 2 enzyme mechanism. There's also strand passage. Now, so we go back to this slide. So you can see that type 1 and type 2 can do these reactions, can relax or supercoiled DNA. Type 1A enzymes can do this because they require single-stranded DNA for activity, so they can not single-stranded DNA rings. They can do this. Now, they can also do the type 1A enzymes can also do these reactions, provided there's a nick or a gap on the DNA. So if there's a single-stranded region on the DNA, they can do these reactions. And remember that because we'll go back to that at the end of the talk. Okay. Now, type 1A can do these ones. Type 1B cannot do it because they're swiveling enzymes, so they don't pass strands. Okay. So the ones that can pass strands can do this kind of reactions. In particular, catenation if you have a nick or a gap. Okay. Now, many years ago, we solved the structure of E. coli topo 1, the strand passage domain of topo 1. And I won't go much into the structure, the details, except to mention a couple of things. It's made of 4 domains, okay? And there's one domain here, one domain here. The active site is at the interface of 2 domains. It's here, the active site. And there's this lid from these other 2 domains. And there's a central hole. So it's a toroidal protein with a central hole here. And you can, in order to enter or exit the hole, you have to lift this domain. And then you have a gate to the central hole. Okay? And it turns out that's the way these enzymes work. You have to lift these domains to enter here. So the active site is at the interface of the domains. It's actually hidden. Okay, it's not accessible to the active site. But if you lift these ones, you actually can enter the active site and go in and out of the hole. Okay? We have now solved the structure of other topoisomerases. This E. coli topo 1, this E. coli topo 3, and I'll go back to the difference between topo 3 and topo 1 in a few minutes. Okay, they are very, very similar enzymes. This is strand passage domain of topo 1. The intact topo 3 is missing a few amino acids at the end that are disordered. A major difference in topo 3 is this presence of this little loop called the decatenation loop, and I'll go back to that at the end. Okay, but overall they look very, very similar. We have also solved the structure with DNA bound to it, single-stranded DNA bound to it. Other people have solved it in covalent intermediates. Okay, but you can see it here with DNA bound. This topo 1 with DNA, and it's just partially bound in this DNA binding group. This topo 3 with DNA is completely bound, entering the active site. And if you look carefully, you'll see that this domain has dislocated and moved on the side. So the active site is formed when the DNA enters. So the binding of the DNA creates the active site, and then the DNA can enter there. It can be cleaved. Okay. Now, what happens after that, we don't know. I mean, we don't have structures after that. We have tried for many years to get a structure of the next group of intermediates, but it has been tricky and probably because they are transient intermediates, they are unstable intermediates. But one intermediate that we managed to get many years ago again is this. If you look at the top domain here, these 2 top domains, okay, this from topo 1, these 2 top domains, okay, it turns out that there are 2 conformations. In the same crystal structure, we could see 2 conformations. This conformation, which corresponds to that, and another conformation where it actually breaks here and opens up. And you can see here, as it rotates, that it sort of opens up. Okay. Now, based on all these structures and many, many biochemical experiments by us and by others, we have come up with a mechanism of how this works. And I won't go into this. Okay, I'll skip this. But this is the mechanism that we propose, similar to what Tony was saying for topo 2. This is the topo 1 mechanism. So you have the enzyme in a closed conformation. It has to be closed, so the DNA cannot just go there and be cleaved. Okay. So it senses single-stranded DNA by binding single-stranded DNA here. Okay. When it binds DNA, it enters the active site. So first it binds, then it forms the active site, it cleaves the DNA, okay, and then we think, we think because we haven't seen the structures, that it opens up, and this is just a cartoon of how we think it could open up. It's not necessarily the way it opens up. Okay, it opens up here, it drags one strand here covalently attached to it, the other one stays here non-covalently attached, and then the other strand can enter the hole. Okay, and once it enters the hole, it closes again. You reform the broken strand, and then you release the strand, and you have passed one strand through the other. Okay? And we know, we have shown biochemically that this opening indeed happens. Okay? We can trap intermediates by trapping DNA in this conformation. Okay? So we know this more or less the cycle. Okay? When we can see how it happens, it changes the linking number steps of 1, and it could keep going on and on this way, or it could just exit and do one single strand passage mechanism. Okay, so that's our general mechanism. It explains relaxation. It explains decatenation. Here's just an example of a double-strand ring and a single-strand ring. You can see how you can have a similar cycle, and it explains that, or you can go from two separate molecules, and you can catenate by going this way. So you need the conditions. You have one single strand and one double strand, or you could have two double-strand rings, one of them with a nick or a gap. Okay. Now, a lot of people here do calculations. Just warn you, this is not based on any calculations. These are extrapolations between actual structures. There's intermediates that we have trapped, and we just came up with these cartoons or movies of how we think the enzyme could work. This may not be right. Okay, in the sense, I warn you that this is based on structures, but the opening here, we think it's going to be that way. It doesn't have to be that way. It could be opening more towards the bottom. In fact, I think it probably opens more by dropping and opening towards the bottom. But the intermediates are correct, and all the series of conformational changes that you see here are based on actual structures. So the molecule is fairly dynamic. When it binds DNA, that's what is missing here. When it binds DNA, all these conformational changes are driven by DNA binding events. This is driven by DNA binding events, and then is the opening event. That's what we still don't know exactly how it happens, but we expect that it's going to be something like this, where these two domains separate. Okay? So we have a fairly good idea of how this enzyme works. Okay? So what we've been doing lately is trying to understand the difference between different topoisomerases. Okay? And in particular, same as what Tony was saying, this is type 1A enzymes, and here we have topo 1, okay, in E. coli. It has the strand passage domain. You saw the structure of that, okay, and it has a large DNA binding domain, has a zinc domain, binds some zinc, and topo 1 protects about 40 base pairs. So it has a big footprint. So it binds to a large region of DNA. Topo 3 has a very small DNA binding domain around here. Okay, it's about 14 kilodaltons, and it protects about 14 base pairs. Okay, so it protects a much smaller piece of DNA. Okay, now Cerevisiae topo 3 and human topo 3 are more similar to E. coli topo 3. I should say, in bacteria, you have E. coli topo 1 and topo 3. In bacteria, you have topo 1 and topo 3. Okay? In eukaryotes and in many archaea, you don't have a topo 1A. You have a topo 1B, but you still have a topo 3. So they do have a topo 1B and a topo 1A. So topo 3 is present in archaea and in eukarya. Okay, so you have these two. Okay, and there are other in archaea, for example, this reverse gyrase, which introduced positive supercoils, and it's a combination of this strand passage domain and what we call a helicase domain. It's not clear that it's a helicase domain, but the combination of these two domains can actually introduce positive supercoils. So this is a strand passage domain. Okay? So what we were interested is to understand what's the difference between these two enzymes. And in vivo, okay, there are differences between the enzymes. Okay? Topo 1 is involved in transcription and replication, and topo 3 in recombination and replication. Okay? And in vitro, topo 1 relaxes negatively supercoiled DNA very, very efficiently, and it can catenate and decatenate molecules inefficiently, provided there's a nick or a gap. Okay? Topo 3 is exactly the opposite. Okay? It relaxes DNA very inefficiently, but it can catenate and decatenate molecules efficiently. So one thing we want to understand is what makes these two topoisomerases different. Okay? And just to show you that, this is a bulk experiment, meaning a gel experiment, and just to illustrate, here's topo 3, topo 1, just relaxation of DNA, varying the amount of enzyme. You can see that topo 1 relaxes DNA pretty well. Okay, topo 3, you need a lot of enzyme, and you don't get into completion. So they do relax DNA differently. So we want to understand the difference between these two enzymes and see, it gives us some clues about their activity in vivo. Okay. So the way we decided to do it is by single molecule approaches. Okay. And I'll explain how the experiment is done. Okay. What we do is a single DNA molecule experiment. What we do is we attach a single DNA molecule. Okay. It's attached to a paramagnetic bead, a latex bead. Okay. One end is attached to the bead, the other end is to a glass slide. Okay. And then we can use a magnet to pull on the DNA to stretch it. And we can also rotate the magnet to introduce supercoils. And we rotate clockwise or anti-clockwise. Anti-clockwise, we can introduce positive or negative supercoiled DNAs. So here's, you rotate this way, okay, you introduce supercoils. Now, what happens when you introduce supercoils into the DNA, okay? That first it just starts unwinding the DNA, but there's a transition when it starts forming plectonyms. And you start forming plectonyms, and when you form plectonyms, okay, the DNA is shortened. So what you see is the bead goes up. Okay? So you are tracking the position of the bead. You cannot see the DNA, you can see the bead. The bead is large, it's about 1.5 microns. So you can track the position of the bead, and now it goes up. Okay? So just to show you what an experiment, a calibration curve would look like. Okay? If you do it at low force, okay, if you are here with your DNA, you stretch it, here it's at 0.2 piconewtons, you can introduce positive supercoils, okay, the DNA shortens, at this position is the buckling transition, it starts shortened, it's linear here, you can go negative supercoil, and the same thing happens, okay. Now, at low forces, the curves are symmetric, meaning so you can introduce positive or negative supercoils, and the length changes because you introduce plectonyms. At higher forces, negative supercoils start melting the DNA. And when you pull at high force, the DNA is melted, so you don't see any increase or decrease in length, which means that you can only do negatively supercoiled experiments at low forces, okay? So you can only do your negative supercoiled experiments below 1 piconewton, so around here, 0.7 piconewtons is what we normally use, okay? But you can do experiments with positively supercoiled DNA at very high forces. So why would you like to do high forces? Because it reduces the noise. The noise that you see in your data is given by thermal motion. The bead is moving by thermal motion, and that's your noise. The more you pull on it, you reduce the thermal motion. So the higher the force, the better for the data quality. Okay. Now the other thing we did is we used two different substrates. As I said, we don't want to use only negatively supercoiled DNA because of the differences at high force, but we could use not just regular double-stranded DNA, okay, and we can use negatively supercoiled DNA for that, okay. But the data is noisy. We have a limited force range, okay, and we don't know where the single-stranded regions are appearing. We don't control the number of single-stranded regions are appearing. Alternatively, in this, what we normally do is we can use DNA where we introduce single-stranded regions. In this case, a 12 or 27 base pair bulge, or a mismatch where you have two strands that are mismatched. Okay, and this has done before for Topo 1. We were not the first ones to do this one. And then you can use positive supercoiled DNA, because then you can actually use positive supercoiled DNA, and topoisomerases would recognize these things. So the advantage is that you can use higher forces, less noisy. You have stable positive supercoils. Okay, you can control the size of the DNA regions. You can control the type of the DNA region. You can control even where you have the DNA region, and you know how many single-stranded DNA regions there are. So there's only one single-stranded DNA region. So we know it's only one topoisomerase acting on only one DNA molecule. Okay? In this case, there could be two topoisomerases acting at once. Very unlikely given the concentrations that we use, but in principle it could happen. Okay. So now, this is just to show you some quality control. Okay, these are the 27, 12 base pairs bulges, mismatches, okay, and you can see that the calibration curves, they just look like double-stranded DNA. So introducing these single-stranded regions doesn't really alter the behavior of the DNA, so we can do our experiments. And let me show you typical experiments. Okay, now here's an experiment with at 0.7 piconewtons with regular DNA, negative supercoils with Topo 1, and you see we introduce the supercoils, and it relaxes the supercoils. Okay, Topo 2, we introduce the supercoils, and it relaxes the supercoils. Okay, we can do it with the bulges. Okay, we can do it at much higher force with the bulges, so you can see right away the difference in the noise level. Okay, and you can see here we introduce the supercoils, and it relaxes the supercoils. Okay, we can also do it with mismatch substrate. The mismatch at higher forces doesn't behave very well. It has a hard time, and we think because if you pull hard enough, these mismatches start looking like regular strands. So you have to pull like gently so that you can still look like single-stranded DNA regions, but you can see here how they relax the DNA. Okay. Now, if you notice, okay, it doesn't relax steadily. It relaxes in steps. And to characterize the topoisomers, we parameterize in different ways. So let me explain here. So we introduce first our negative supercos or positive supercos, and what happens there is that there's a lag time here before we see any activity. Okay? So this is just, and we call it time lag, the initial time lag or primary time lag. So you wait, this is the time that you have to wait from the time you start supercoded the DNA until you see the first event. And then it starts relaxing the DNA. Okay? And it can relax many turns at once. It doesn't mean that it does more than one step in a cycle. It means that the time resolution of our instrument is not high enough. Okay? We collect about 40 frames per second, so it's going faster than that, so we don't see individual steps. With some substrates we do, but not with this is with negative supercoded DNA. So you see it relaxes, then it takes another pause, it waits here for a while, and then it relaxes again, and then it's done. Okay? So we call this the initial time lag. This is the number of turns it relaxes. The rate is just going to be the slope of this part, which is the number of turns, and divided by the time it took to do it. There's a secondary time lag, and then the total rate is what it takes from the instant we finished introducing supercodes to when we decided it has finished relaxing the DNA. That's our total time. Okay, so we do all this. So we collect lots and lots of single molecule experiments for different substrates at different forces, and then we look at all these parameters. Okay, and this just to show you what happens here. The first thing I'll talk about is the initial time lag. Okay, and this is just the average values. Okay, and these come from exponential fits of the distributions. So it's yes.
Is this a processive or a distributive reaction?
It's a processive. Okay.
So it's the same enzyme that's bound.
It's the same as far as we know. Okay. Well, everything indicates that it's the same enzyme. Well, I cannot assure you 100% that one of them didn't float away and another one replaced it immediately. But given the concentration, given the times, we think it's the same enzyme. And we're doing the experiment to actually show that it really is the same enzyme by looking at the enzyme at the same time. But right now, it's based on the assumption that it's the same enzyme. Okay. But we think it is. Okay. When you look at the time lag, you see one thing right away. And as I said, these are the mean values from many, many experiments. Okay, that's why you want to look in single molecule experiments, not at individual events, but at the average of the many events. The time lags for topo 1 are very short. Okay, so it means topo 1 starts acting on the supercoiled substrate right away. The time lags for topo 3 are very long. Okay, so topo 3 takes a long time to start acting on DNA. And that's true regardless of what substrate we use. So it's true of the bulges, it's true of the bulges. In fact, in this substrate, I put here a dot. It's because these are probably not very good values, these values, because it took so long that we stopped the experiment. Okay, we waited long, like over 15 minutes, and nothing happens, and we decided, okay, the enzyme is either dead or it's going to take forever. So these are very few events. Okay, well, not that few, but not as many as these ones. So we think this is an underestimate. Okay, but still you can see that it's much longer for topo 3. So topo 3 always waits longer before it starts acting. Okay. And that's true also of the secondary time lag. Topo 3 always waits longer. Okay, which argues a bit about being the same enzyme. Okay, that the time lags are the same. Okay, so it's not that we're waiting an event. Okay. Same thing. Topo 1 waits. The secondary time lags are short. Topo 3, the secondary time lags are long. Now, this is more interesting. Okay, what happens to the relaxation rate? And that's the slope of this. Okay, just of individual events. And you can see here that just shows the distribution. So you see we have a very nice exponential distribution of events. Okay, we take the mean of the distribution. Okay, and you can see here that the opposite is happening. Topo 1 actually relaxes DNA pretty poorly. Okay, and topo 3 relaxes DNA very, very fast. Okay, so topo 3 relaxes DNA very fast, and this is topo 3 data. You can see the slopes are very, very high. So once it starts relaxing, it goes very fast, and that's true of every single substrate. Okay, so topo 3 wins. It's a very fast relaxation. It's very processive. It does many turns at once, and it does it very fast. But what happens when you look at the total relaxation rate, meaning going from here all the way to there? Okay. What happens there, and that's a combination of the time lags together with the relaxation rates, that when you combine that, topo 1 is a more efficient enzyme. Okay. It's the time lag that is making the difference. Topo 1 relaxes faster than topo 3. And that's true of every single substrate. That's again the distribution. So you can see here that topo 1 is a much better enzyme at that. Okay. So let's look at the numbers quickly, and you don't need to go through all this table of numbers. These are all the characterizations of all of them, just to show you that the initial time lag, it's always shorter for topo 1, no matter what the substrate is. Okay. The secondary time lag always shorter for topo 1. Okay. The number of terms removed, how processive it is, it's about the same. Okay. The relaxation rate per run, just one run, how fast it goes. Topo 3 is much faster, 40 times faster than topo 1. Okay. But the relaxation rate, the total relaxation rate, topo 1 wins. Okay. So what you see here is just a summary of two of the substrates, just for comparison, but just pay attention to the negatively speckled DNA. Okay. The relaxation rate is low, it's fast. The initial and secondary time lag are short, these are long. The total relaxation rate is 0.9 and 0.2. So topo 1 on bulk experiments should appear to be a much faster enzyme, and it's true. I mean, if you look at the bulk experiments, topo 1 is a much faster enzyme, and topo 3 is slower. But the reason is not that just topo 3 works slowly. In fact, topo 3 works very, very fast. It's a very efficient enzyme at removing supercoils. It just waits a long time before it does it. So it sits there waiting for the correct substrate before it acts on it. Okay, that's something that you would not be able to see from a bulk experiment. The single molecule experiment reveals it right away. So what we think is happening here, okay, is that we have one of these examples of the tortoise and the hare. So topo 1 is the tortoise. It just relaxes DNA slowly and steadily. When you say go, it starts going and goes all the way to the end. Topo 3 is here, runs very, very fast, but it's taking lots of naps. Okay, so by the time when it tries to catch us up, topo 1 has already relaxed the DNA. So it's one of those cases that you can see the difference now in the single molecule experiment. Now, does it make sense in terms of what we expect them to do? I think it does. Okay, so this is just this partial summary. Topo 3 relaxes DNA efficiently in fast processing runs, but with long pauses, while topo 1 is in slower runs, but with shorter pauses. Okay. And the combination of these two characteristics makes topo 1 have an overall faster rate. Okay. Now, I think that makes sense in terms of their function, because now topo 1, as you heard before, is involved in just housekeeping functions in E. coli or in bacteria. Okay. So it's an enzyme that can monitor and alter the DNA constantly. So if something happens, transcription, something happens, it right away takes care of it. Okay. It does it slowly, but it does it right away. Topo 3, on the other hand, okay, acts very fast, but it waits a long time. Okay, so it may be waiting for special substrates. So just on certain substrates, it can relax them very, very fast. But regular supercoiled DNA, it does it slowly. So probably in a situation where you have both proteins present, okay, topo 1 will relax the DNA, do all the housekeeping job, and topo 3 is not. Before it starts, it's already done. Okay, by topo 1. So I think that makes sense in terms of their function. Now, about 10 minutes. Yeah, that's about right. Okay, so good. Okay, so let me just tell you another half of this story, and this half is already just came out last month, but the second half is still work that we're still doing, so it's not as complete. Okay, but that's looking at the other aspect of the reaction. That's the decatenation reaction. And we know from the first time when Russell DeGate found first topo 3 as a decatenating enzyme, that topo 3 is a very, very potent decatenating enzyme. It's just decatenating rings with a nick. Okay, topo 3 can decatenate and topo 1 can do a poor job. Okay, so since then we know that topo 3 is involved in recombination, it's involved in replication, and that what people think it does is resolve this intermediate. It helps resolve this recombination intermediate, single-stranded crossovers intermediate. So it does this. Okay, so that's one thing that topo 3 can do. Okay, other things that topo 3 can do, okay, is that it can resolve replication intermediates and we know that most of the time it's topo 4, but under certain circumstances, you can imagine that topo 3 could do it, and they show that you could do it under certain circumstances. That if you have single-stranded intermediates, you can do this reaction, decatenating those two by resolving these single-stranded intermediates. Okay, as I said, it probably goes this pathway, but you could do that one. Okay, so there's another topoisomerase that could do that one. Okay, but there's another reaction that actually caught our attention a few years ago, and particularly recently, because we thought we could actually test this one. And that's one that it was noticed by Steve Kowalczykowski, and it's that in the presence of REC-Q, which is a helicase. Okay, so if you have REC-Q on topo 3, you can catenate and decatenate double-stranded molecules. So if you have double-stranded molecules and REC-Q, you can decatenate it. And this has been shown to be important not only in E. coli. The equivalent of REC-Q in humans is the Bloom helicase, and there's a topo 3 in humans. And people have proposed that the combination of, we know that they interact, REC-Q and topo 3 or Bloom helicase and topo 3 interact. Okay, they actually interact and they work together. So people think that it's an important reaction. Okay, I won't go into all the details about it, but just to say that if you have REC-Q as a helicase, okay, and you can unwind the region of DNA, okay, and then you have a topo 3 could clip here, pass the two strands into here, form a hemicatenate. Okay, so this is a hemicatenate. Okay, so one double strand is inside the two other strands. Then you have the same or a different molecule clip here, pass the other through the other strands, and then you can catenate two double-stranded rings. Okay, or you can decatenate two double-stranded rings. So you can do this reaction with a combination of REC-Q and topo 3. Okay, and there's a lot now about what happens when you don't have REC-Q and all these kind of things, but I won't go into that. Okay. One thing that also caught our attention is that from Steve Kowalczykowski, if you look at, they look by AFM at those catenated rings, and they look very nice. Here you can see the catenated. You can see how they look. So we look at that and say, well, maybe we can use our substrates to start to study this reaction, the decatenation reaction. Okay, but we don't want to have REC-Q because we don't have a way of controlling where the single strand is going to be. Instead, we could use our substrates, okay, that we already have, and we have to make these catenated molecules. Okay, so the way we mimic catenated molecules is by making braids. Okay, so in this case, we put two double-strand molecules attached to our bead. Okay, and now we turn the bead, they braid each other. Okay, so you braid the molecules by having two molecules in a bead. Okay, so what do we think they look like? We think they look like that. So when you form crossover points, it's a braid, and now we have two DNA strands going together, and we think if we introduce here like a bulge, a single strand bulge, maybe a topo 3 or a topo 1A can catenate and decatenate these molecules, because this would sort of mimic what REC-Q is supposed to do, melt the DNA. So you can use this as a substrate for topo 3, pass one strand, and then pass the other one. Now, every time you decatenate these molecules, you need to do two strand passage events. Okay, and we only have one single stranded region, so we still don't know how that could work. So these are the braids that we use, and this is the way the braids behave. And just to remind you, a braid, imagine that you're in a swing, and instead of swinging, you actually turn around, and you see children do that. Okay, so when you turn around, and then the swing goes up. It's the same thing here. Okay, if you turn, the bead goes up, and it's symmetric. Either you go negative or positive supercoil, and you see this little peak here, which is characteristic when you have a braid, and that's because the last step of your braid, you go down a lot. Okay, so that's the way that braids look like. Now, the braids we were using, okay, even for intact DNA, we only attach them by one strand. And the reason is that we don't want to introduce supercoil. So if you attach them by both strands and turn them, even though they're braided, they also get supercoil. So we only attach them by one strand. Okay, or we can nick them, okay, to prevent them to supercoil. Okay, so we can have intact DNA with nicks, intact DNA without nicks but attached by only one strand, or nick DNA with a bulge. So we did all those substrates, and here's just some data. What happens when you show intact DNA, braided intact DNA? Okay, there's no activity. Okay, and that's what you expect. You look here, and you look here, and say, we're waiting for a long time and absolutely no activity. So topo 1 or topo 3, they cannot unbraid intact DNA, and that's what we expect. How about with nick DNA? And these are single nicks, okay? If we introduce single nicks, every now and then we see what maybe it's an event, okay? This is an event, but most of the time, there are no events. So nick DNA is not a very good substrate. And we know that. The ones that work well are gapped DNA molecules, where there's a huge single-strand regions. So nick, when you just have one small nick, they're not very good, okay? So nick DNA doesn't work very well. So how about with the bulges? Okay? This is decatenation activity with a 27 base pair bulge on DNA. And now what you are seeing is that you start seeing activity, okay? This is a better example. You can see topo 3, okay? It unbraids, waits, unbraids, waits, okay? And then we introduce more braids and do it again and again, okay? Now we have done this experiment a few times, not too many times, and we're still analyzing the data, but we have some preliminary results already that seem to make sense, okay? When we compare now how many terms it relaxes, well, it unbraids in one event, shouldn't be DLK, it should be change in braids, okay? It's about 2 braids in every time we see an event, okay? The rate is slightly faster for topo 3, but not that much faster. But the time lag there, now it's, the time lag for topo 1 is much longer than the time lag for topo 3, okay? So we look now at the numbers, okay? And you can see here the time lag of topo 1 before you see an event, it's about 94 seconds, on average 22, okay? So it's a bit over 4 times slower for topo 1. The number of turns removed is about the same, the rate is comparable, okay? So the time lag is smaller or shorter for topo 3, the number of turns on the rate are similar for topo 1 and topo 3, okay? If you compare that with same substrate, but in the super cold when it's relaxing, okay? The time lags for topo 1 for super cold DNA are short, and the time lags for topo 3 are long. It's reversed here, okay? Topo 1 and topo 3, the number of turns is about the same, okay? The rates are, well, comparable, okay, not the same, but comparable. So it's the time lag that is making the difference. This is the part that makes the difference. And now in the case of catenated DNA, okay, topo 3 has shorter time lags and has a faster rate. So we haven't calculated it, but we don't need to calculate to know that the overall rate is much faster for topo 3 than for topo 1. So topo 3 decatenates DNA much faster by having shorter time lags, even with comparable rates of decatenation reaction, okay? So that seems to explain, at least to some level, what's the difference between the two of them. The intermolecular reactions are much faster for topo 1 because it waits shorter periods of time before it starts. The intramolecular. For the intermolecular, topo 3 is a much better enzyme. Okay, so what's happening at the level of the structure? We don't know exactly what's happening, but one difference is that topo 1s have as I said, big, large here C-terminal domain that binds DNA. This is from Thermotoga maritima topo 1, the only one that we know the whole structure. You see here, large domain here. Topo 3 has a small domain, okay? But topo 3 also has this decatenation loop on the other side of the molecule, okay? And that decatenation loop, we know it does something. It's not just a loop hanging out there. We showed many years ago that if you remove that loop, the topo 3 decatenation loop here, you lose decatenation activity. Relaxation is much worse, okay? So that decatenation loop is doing something, but we don't know what it's doing. So we'll have to work out what that loop is doing. But one thing that could be happening is that topo 1 with a much larger footprint engages the DNA initially much better when it's the same, when it's an intramolecular reaction. It engages the DNA and it prepares the DNA. Remember, the protein and the DNA probably have to change conformation to start relaxing. So it engages the DNA, but then it's a slow enzyme. Okay? The strand passage event is slow, and maybe it's because with such a large footprint, the DNA cannot keep moving in and out easily. Topo 3, on the other hand, has a small DNA binding domain. Okay? So it may take a long time before it actually engages the DNA and changes the conformation of the DNA, prepares the DNA for the reaction. But once it does that, with a smaller DNA binding domain, it may be able to move it around faster. So it can do strand passages event much faster. And in some way, that decatenation loop is helping. Okay. Notice that this decatenation loop is not part of the domain that is moving, or that we think is moving, but it still allows the DNA to go in and out much faster. Okay. And in some way, it must help in the intermolecular case, but we don't know how. But we think that we're starting to get some clues of what makes these two enzymes. And I'll just finish by saying that what we have shown recently is then that on a supercoiled substrate, topo1 is a much better enzyme because it has shorter pulses, not because it relaxes. The strand passage events are not faster, they're actually slower. While topo3, okay, although it can do strand passage very fast, okay, it's slow and starting, okay, it pauses a long time. But for intermolecular reactions, topo3 appears to be faster at passing strands and faster at starting the reaction. And these characteristics, these two characteristics seem to explain the mechanism of the enzymes, or the difference in mechanism. They use the same mechanism, just in a different way. Okay? And I'll stop there and just thank the people that have been working on this. And there's a long list of people that work on topo1 in the lab on the structure, many other studies, but the most recent work is a student that is just about to finish, Ksenia Terekova, who did all the single molecule work. And the single molecule work has been a collaboration with John Marco, who is my neighbor at Northwestern. Thank you very much.
Thank you, Alfonso. Dorothy.
I just had a quick question about the single molecule data you showed with the braids and the 27 base pair bulge.
Yes.
I was expecting the length to increase monotonically as time went by, and yet you had these steps going up and down. Did I misread the graph, or is there something going on about the braiding?
With the topo presence, or just the calibration?
No, with the topo present, yeah.
With topo present, but we don't have the time resolution for it to go to see enough. Okay, so what happens is that the events are so fast that we just see like this almost discontinuous jumps, and it's just because we don't have the time resolution. We're doing
it just looked like the jumps were going in both directions.
That's misleading.
Oh.
It's because we do an event, it decatenates, we get bored,
right,
and we introduce more
I have more tolerance for boredom. Thank you.
So I think this is so exciting because it's been an interesting question for a long time, and I know you keep peeling away parts of the onion skin. But I want to overlay something on this, is that it's very important that there's exactly one topo3 molecule per E. coli cell, where there are up to 10,000 topo1s per E. coli cell. And we looked really, really hard in vivo in a cell to see if topo3 ever made a contribution to decatenation of replication intermediates. And the answer is absolutely, for sure, never. Even when we overexpress it, we try to make it never. But it's more important for that recombination resolution, because one step in the wrong direction is death to the cell, right? So people are so excited about decatenation or replication. There's a million ways to get out of that problem.
Topo4 does.
Yeah,
I mean, we know that
Stochastic
Topo4
nerve.
does that. Okay.
But one false step by topo3 is death for the cell. So I think that your data completely explain those.
I don't know if it completely explains, but it helps explain. But then, yes, I mean, it can do many other things.
Pause, and act when you have to.
But I think you're right in that the important reaction for topo3 is the RECQ topo3 collaboration, which also happens not only in bacteria, it happens in all organisms.
A question about the setup of the experiment. So you twist the molecule, and then you put many molecules of topoisomerases or whatever. Are you controlling the concentration of these, the concentration of these molecules around the chain to see if the reaction is at the same time, or there are specific holes along the chain where the reaction is not happening? Okay, so this could explain also these big steps.
No, well, two things. The way we do the experiments is that we flow, we put the DNA, we flow buffer, then we flow protein, then we supercoil the DNA. Okay, so there's protein present when we supercoil the DNA. Okay, the concentration is the nanomolar range. So if you make the back of the envelope calculation, there should be about 1 molecule per DNA molecule. So there's very, very low concentration. And the other thing is that for the positively supercoiled substrate, which is for that reason I think it's an important control, there's only one binding site. topos won't bind, and topo 1A only will bind to single-stranded regions. They won't bind anywhere else. So in those cases, there's only one DNA molecule, there's only one protein molecule active.
Okay.
The only question that we haven't answered is what Steve was saying, is like, is it the same molecule active? Okay. It could be that whenever it stops, a molecule comes off and another one goes on, that the lag times are waiting. We don't think that's the case given the concentrations. Given such a low concentration, we would expect a much longer waiting time between events. And the fact that the events, the waiting times are comparable makes us believe that it's the same molecule, but we still have to show.
Okay. Thank you.
All right, thank you. I think we better move on. Time's getting on. So thank you, Alfonso.